Hello, welcome to another new class of human physiology. Hopefully you are enjoying different classes of human physiology. In the last few classes we discussed about different endocrine glands. We also discussed about about glucose homeostasis, and also we further said that if any irregulation of the glucose homeostasis happens due to various reasons, the condition of hyperglycemia, which is a deadly disease which is called diabetes. We briefly discussed different therapies of the diabetes and their advantages and disadvantages. In today's class, we will discuss about a modern approach of therapy for diabetes, mostly for the type 1 diabetes, which is like the cell based therapy. Briefly also we will discuss how we can develop artificial pancreas. So this would be very interesting class. Let's stay with it. So what would be different contents which will be covered in this class? We will discuss about what is glucose homeostasis. We will see if any irregulation or dysregulation happens in the glucose homeostasis, how diabetes develop. Mostly we will focus on the type 1 diabetes. Then we will see briefly discuss about different limitations of the current therapies. After that we will introduce you to the cell based therapy, and we will discuss about how cell based therapies can be used for type 1 diabetes. Then we will introduce a new concept of fibrosis, and we will also discuss that what are the challenges related to cell based therapy due to fibrosis. Then we will discuss about immunoisolation and how alginate based polymer or other natural biopolymer can be used for immunoisolation of the cells. Lastly, we will discuss about an interesting component of vascularization and why vascularization might be helpful and needed for cell based therapy. So this would be a very interesting, maybe little bit longer class. So let's enjoy. So you remember in last few classes we already discussed about what is glucose homeostasis, and the primary thing is our pancreas has mostly two important cells, right? One is the alpha cell and then second is the beta cell, right? And what we said, the beta cells, it secretes the insulin, right? And the alpha cells, it secretes glucagon. And how they maintain glucose homeostasis? Whenever we eat any food, those foods basically after the digestion breaks down to carbohydrates and glucose. Eventually the beta islet cells from the pancreas, they secrete the insulin. They help in terms of carrying those glucose with it, and eventually it distributes those glucose molecules towards various cells and organs of our body. In case any excess glucose stays in the body, what this insulin does? This insulin basically converts the glucose to the glycogen and it's stored in the liver. So basically any excess glucose, it converts from glucose to the glycogen and it stores in the liver. And now whenever there is a prolonged fasting state, that means the glucose level goes down, that time what will happen? The alpha cells of the pancreas will secrete another hormone which is called glucagon. This glucagon will further convert glycogen back to the glucose, and then this glucose will anyway get distributed to the body, right? So in this way insulin and glucagon basically maintains the glucose homeostasis of our body. But what happens in case of any irregulation, if in case this pancreatic islet cells get damaged by anything, or maybe insulin resistance happen, we experience a persistent high glucose condition which is called hyperglycemia. Prolonged hyperglycemia state which becomes a disease condition which is also called as diabetes. Now diabetes, as you remember, we said there is two categories of diabetes. One is type 1 and then second is type 2. Both of them we discussed in our last class also, but in this class we will only focus about type 1 diabetes. So as we said, in case of type 1 diabetes, our body generates an autoimmune reaction, and it mostly happens very early, about less than 10 years of age in the kid. And our hyperactive immune cells, including hyperactive T cells, basically they destroy all our pancreatic beta islet cells. So all the pancreatic beta cells which gets destroyed by autoimmune system, causing deficiency in formation of the insulin. So basically there is almost no insulin production from those pancreatic beta islet cell in the type 1 diabetes patient. And as you can see, almost 5 lakhs of children which are less than 15 years of age are like suffering from diabetes, type 1 diabetes worldwide. About like 11,000 are from India. And as you see in the figure that in case of type 1 diabetes, because there is no insulin in the blood, they are unable to supply the glucose inside of our cell. So basically this glucose is completely cut off to different type of cells, including our brain cells. And as you know, like if glucoses are unable to like get transport towards various parts of our body, the blood glucose level will go high and it will create like a condition called like hyperglycemia. And due to lack of glucose in the brain tissue, the hypoglycemic condition also can generate in the brain, causing like instant death. So basically type 1 diabetes is a deadly autoimmune disorder. What are some general therapies of type 1 diabetes? We already said like of course like the most common one, like you can give insulin. So insulin based therapies can be used, but the problem is like this is like a continuous administration is needed, because in case of type 2 diabetes people or the patient still has some active beta pancreatic cells left. But in cases of type 1 diabetes, there are no active like pancreatic beta islet cells left in the body. So you can understand a kid that doesn't have any pancreatic beta islet cell, whenever he or she eats any food, he needs an insulin jab or insulin injection. So it becomes very painful. The parents has to continuously administer their like glucose level, and they need to probably inject the insulin whenever required. And sometimes it happens like parents can judge it wrong, and if the insulin level or insulin injection gets more in the body, and then the patient can experience also the condition of hypoglycemia or the brain coma. So this is a significant issue, and persistent insulin injection also can slowly develop insulin resistance also in the body. In that case, insulin will not be able to properly act and bind to the receptor. The second most important therapy that can happen, which is like the organ transplant or the pancreatic transplant. But problem is like how many donors are actually there, right? Even in the US, the government data says like not more than 10 to 20 pancreatic organ donation actually happens. So basically you can understand, although this can be effective, but the numbers are very low. Apart from that, there is a risk of like organ rejection by the immune activity or the host immune response. So I will just introduce this term here, the host immune response, which is basically the immune response towards any foreign object, in this case the transplanted pancreas. So if the host immune response kind of detect the transplanted pancreas as the as a harmful object or the harmful foreign body, it will basically destroy it. And to prevent against like the foreign body response, doctors generally prescribe like the steroids or immunosuppressive agents. And as you know, like long term use of immunosuppressants are not good for your body, because it also compromises with your body's immunity. So as you can see, like there are challenges with the insulin therapy, there are also challenges with like the organ donation. Then what can be like the alternative approach or the modern therapy that is basically being explored, including our lab. So in this slide we will discuss even like also different like basic physiology, but also will give you like different research aspect also. So it might be little bit extending. If you have any questions, please ask us, right? So this would be kind of a research oriented lecture today. So what can be modern approach, right? As we said, pancreatic islets can be eventually given to the patient, right? But problem is you don't have sufficient pancreatic islet. Why? Because there is not enough like awareness to donate our pancreas. So either we are unable to completely kind of get the whole pancreas, or even like some amount of pancreatic islet cells. And another problem is human pancreatic islet cells are very difficult to culture. Basically they do not regenerate. But there are different strategies nowadays they have developed to kind of generate the stem cell derived pancreatic islet cells that can be cultured in the like lab condition, but that is still highly costly. So the approach that can be taken is like use of xenogenic. So the use of xenogenic islets that either can be taken from like dogs or like monkey or can be taken from pigs. So basically we can take the pancreatic islets from other source, right, other organisms, and then we can transplant that in the body. But there are certain issues, right? So if we just take the pancreatic beta islet cells from this body and inject, right, and inject to our body, what will happen? So in the last slide only I said that our body will generate a host immune response. And what will happen, because these organisms are different than the human body, once these cells are transplanted or implanted to the body, our body will immediately detect this as a foreign object and a harmful object and immediately trigger an immune response to destroy them. So just by injecting this genogenic type of pancreatic beta islet cells to the type 1 diabetic patient is not a solution, because they will not be able to survive more than 24 to 48 hours in the body. So what can be done? We can create like a immunoisolation or kind of a protective barrier. So you can see like in the cell based therapy mostly acts with like therapeutic use of a cells, mostly like either human cells or different type of cells, and these cells are generally given inside of a protective barrier. So what this protective barrier does? This protective barrier basically prevents the immune cells to come inside. So basically it prevents the immune cells to come inside. It also has like proper pores, or these are like these have like proper porosity, so that all the nutrients and oxygens can come in. So basically what it allows inside, it allows like oxygen, nutrients, but they don't allow the immune cells, right? And it also, because it has the beta islet cell, if you consider, so this beta islet cell will release what? Insulin. So this insulin can also comes out, this insulin can comes out, right? And all different type of like B complex proteins, they will not be able to go inside. So basically we can create a protective barrier, and we can use like the human or different other species cells to create like a cell based therapeutics that can be a modernized therapy for type 1 diabetes. But as I said, even if we create like a protective barrier around these cells, this whole system, for example, if this is like a capsule and we have like the beta islet cells inside, this whole system is still like a foreign object, right? This whole system is still like a foreign body or foreign object to our body, right? So even though the immune cells, for example, like T cells, macrophage, right, NK cells, so these different type of immune cells, you remember we discussed in our blood class and immune system class, even if these different type of immune cells cannot penetrate the capsules, even if they cannot go inside, what happens is like body has another secondary immune response system, and that gets mostly triggered by the macrophage. So the secondary host immune response get triggered by the largest immune cells of our body, which is macrophage. So in the primary immune system, if the T cells, NK cells, they cannot penetrate and destroy the inner cells, the macrophage cells will basically come close to this object, and if they detect that this is a harmful foreign object, and of course the object is so bigger than the macrophage cells that macrophage cells will not be able to chew it or destroy it. But what it can do, it can create a foreign body response, and by creating a foreign body response, it will basically create a response of inflammation and eventually fibrosis formation. Okay, what is basically fibrosis? Fibrosis is basically like combination of like different like fibroblast cells and collagen, right? So what we said, like it will create a response of fibrosis, which basically create a thick tissue layer which is made of collagen, and fibroblast cell and fibroblast cells, fibroblast and myofibroblast cells. So this thick layer, thick tissue layer will eventually completely coat that whole system. So you can see in the system, these whole cells and the capsules will be completely covered by this fibrotic tissue layer. And as you can understand that if this complete thick layer of the fibrosis covered the capsules along with the cell, what will happen? The nutrients, the oxygens will not able to go. So the oxygen will not able to go inside, the nutrients will not able to go inside, and even the beta islet cells will not able to pass the hormones, right? Hormones like insulin or even the other toxic metabolites. So in this way the cells inside will die, right? So even though we can create a immunoisolation layer by a like a proper polymer or capsulation system, by a proper layer of the capsulation system, fibrosis can eventually hamper the progress. Okay, hopefully you understood that. Then there are different strategies to prevent fibrosis. Okay, so scientists and researchers are studying since last several years to see how to mitigate like the fibrotic response. So there are like different physical properties. For example, we can change the surface topography, right? So basically we can create rough surface or smooth surface, and mostly if we create smooth surface, it has been seen that smooth surfaces trigger lower fibrosis. So a smooth surface we can create to lower the fibrotic response. And then we can change the size and shape. In terms of size, it has been seen that if the size becomes too small, it creates like a significant fibrotic response. So we can basically increase the material size as much we can to low down the fibrotic response. Okay. Then there are options of different delivering biological reagents also. For example, different anti-inflammatory like cytokines. So anti-inflammatory cytokines like IL-10. Then we can also give like various steroids, right? Various steroids, immunosuppressants. But as you can know, like these all reagents can actually reduce our overall immunity in the body. So in the systemic run, these biological reagents are not good because it can create lot of like immune kind of cascade or immune toxicity, and long term immunity can be a challenge. So the third handle or the third component, which is mostly interesting, why this is needed? Because this physical barrier or the change of physical property, although it can manipulate the fibrotic response, but these are not significant. These are not significant and long term. So the challenge is these are not significant and they cannot prevent long term fibrosis. So basically they cannot prevent long term fibrosis. Okay. The biological factors, of course, they can prevent long term fibrosis, although they can prevent long term fibrosis, but problem is, as I said, because they are biological factor, they can create different immune reaction, immune toxicity, and long term use of immune suppressants are not good for our body. So this third handle, which is in the middle, is highly interesting, use like the chemical modification or attaching various chemical molecules to this on top of this capsules or top of this device. Okay. So these molecules can be deuterionic molecules, these molecules can be small molecules. So by coating or conjugating the different type of capsules or different type of device with the small molecules using chemical modification, basically can actually reduce down the response of the fibrosis. Okay, so this is basically use of chemical moieties to reduce down fibrosis, so that these capsules which are carrying the therapeutic beta islet cells can stay in the body for longer term and can produce like insulin that can correct like hyperglycemic condition. So this is the process which is called basically the immunoisolation, where we can use either a small molecule conjugated like alginate or like different other polymer that can basically isolate like the therapeutic cells from the surrounding environment where active immune cells are there. Also we have to ensure that the fibrotic response are reduced and mitigated, so that our body's host immune response cannot develop like a thick tissue based layer to cut off the whole system from the body. So in this way we can manipulate our body's immune system and create a immune isolated layer, mostly by alginate polymer. So this is already like a FDA approved polymer that is significant in last several years are being used to deliver the therapeutic cells. As you can see, there are several papers what they have showed, if you deliver therapeutic islet cells via using this alginate capsules, almost up to 6 months, normoglycemia level can be maintained, and diabetic level diabetic disease can be temporarily cured. But the problem is, if these alginate capsules are not chemically conjugated to reduce down their fibrosis, eventually it shows the fibrosis and the cells inside of the alginate eventually die, right. So even though they are very good in terms of performing maintaining the normoglycemic level for 6 months, unless these alginate polymers are coated with some active chemical molecule, after 6 months eventually, so after 6 months eventually again the fibrosis response form, and all these implanted beta islet cells, they simply die. So we have to create certain chemical modification around the surface of the capsule, so that the body's host immune response or the fibrotic response can be reduced, and the implant life time or the overall shelf life of the implant can be improved. So in this manner there are certain class of molecule that can be developed, and that is developed by our lab. These are basically triazole type of molecules. So you can see this triple N, so this is called as triazole, and these are different type of small molecules, right. And these triazoles can be developed by a click chemistry based copper catalyzed click chemistry reactions. So this is called as click chemistry reaction, right. And this happens between the cycloaddition between, you can see, alkyne and you can see azide. So basically this is a azide and this is an alkyne. So this is basically a cycloaddition reaction between an azide molecule and an alkyne molecule in presence of copper catalyst, eventually forming cycloadded addition based product, which is a 100% 1,4 isomer, which is also called triazole. And this is highly specific compared to Hughes-Stein reaction. As you can see in the Hughes-Stein reaction, it forms 1,5 isomer along with 1,4 isomer, but in cases of click chemistry reaction it only forms 1,4 isomer with a very high yield. And as you remember, in 2-3 years before only, click chemistry got Nobel awards, right. So in our lab what we developed is we develop a molecular library of several triazole molecules, right. And in that molecular library you can see there is one side which is the hydrophobic component, and another side we created which is the hydrophilic component. So why we are saying it hydrophilic? You can see there are some PEG type of linker. So the PEG is generally hydrophilic in nature, and in the hydrophobic you can see there is this phenyl linker which is mostly hydrophobic in nature, right. So hydrophobic phenyl type of or benzyl type of, right, linker, and in this case there are PEGylated hydrophilic linker, okay. And then we took 50 to 51 different alkyne combination. We had 5 different azide combination, and with permutation and combination you can see we developed about like 211 novel alginate that are conjugated with this type of triazole type of molecules. And you will ask me why we develop like both hydrophobic type of system and hydrophilic type of system. So as you can know, like alikes like. So basically if we want to use this type of small molecule coating in a soft polymer, in a more hydrophilic type of system or polymer, we need hydrophilic type of coating. But in case we want to use this small molecule for a hydrophobic system, for example like a plastic type of system or silicone type of system, we mostly need then hydrophobic type of coating. So basically you just remember this thing like alikes like, okay. So hydrophilic will kind of like hydrophilic system, hydrophobic will like hydrophobic system. So in case of hydrophilic, what different type of substrate we can use for the coating? For example like soft polymers, like natural, it can be mostly natural polymer, and in case of hydrophobic mostly like plastic and synthetic polymers can be used. Okay. So this once we develop like different type of almost see around like 211 new molecule, now what we have to do, we have to test like and identify that which of these 211 molecules are actually best in terms of preventing the fibrosis. So are you getting to the point that initially we develop like a large chemical library, and the goal is now to do like a high throughput screening process to identify that which of the molecule is a lead molecule to prevent fibrosis. So the eventual goal is that what is host immune response we have to reduce the fibrotic process so that the capsules can stay in the body for longer term, surviving all those beta islet cells in the body. So to do this screening, we approach like a genetic barcoding approach. I am not going to discuss in detail because this would then divert from the actual topic that why the genetic barcoding approach has been developed, but just to give you short idea, like in the there is no proper like screening approach in the in vitro for the fibrosis. Like for example, we screen anti-cancer compound or antibacterial compound first in vitro, means like in cultured cells or bacteria, and then once they are cultured, the lead component or the lead compound we test in animal and further in human. But in case of fibrosis, there are no such reliable model. And what happens, like as we have this large number of library of molecule, we need large number of animals to screen this. So unless we develop a high throughput screening approach, screening this number of like excess molecules cannot be done, because nowadays like all the institutes and the regulatory agencies are kind of not allowing us and all the researchers to use too many animals. So we developed a high throughput screening approach using like a genetic barcoding strategy, where we can combine different molecules together, almost like 20 to 40 molecules, almost 20 to 40 different type of 20 to 40 different like this conjugated alginate capsules we can implant inside of the mouse. Right. So implanted all those for 20 to 40 capsules, each has a specific genetic barcoding inside of the mouse, and what we did, basically we kept those for like a month inside of those animal, and after the retrieval we did the microscopic imaging. So as you can see, once we did the microscopic imaging, maximum of those capsules have too much of fibrosis, only very few capsules do not have any fibrosis, only very few capsules do not have any fibrosis. Now we have to identify what are these few capsules, right, inside which came out from the mouse. So how did we identify? Because you remember I said that each of the capsules has a specific genetic barcoding. So by identifying that genetic barcoding, that which genes are present inside of those capsules or which cells are present inside of those capsules, we could backtrack back and identify the lead molecule. So in this way we identified 3 different lead molecule which can prevent fibrosis for long term, which can reduce fibrosis for long term. So now as we identified like reduction of fibrosis in long term, there are like actually 10 like different lead initially we identified from a combination strategy, and all these leads initially, like in this last slide, all these leads were implanted in a capsule size of 1.5 millimeter. Now you remember in like a few slides before I said like if we reduce the size, generally body generates more immune response. So in this case what we did, after the 1 millimeter capsule size, we reduced the capsule size to 0.1 to 0.2 millimeter. So as we reduce the size, what we can expect? So as we reduce the size, we expect that fibrotic response may go more, right? And we want to test all the top 10 lead to see like if we reduce the size, can it still prevent the fibrosis or not? So as you can see, after screening this in individual animal setting, what we identified that is like we identified two different leads, G1A3 and G4A10. They don't have like any fibroblast cells. So you can see like there are different staining we did. So this is alpha SMA, which is like myofibroblast cell. So in the control alginate where there is no small molecule coating, you can see there is so much of myofibroblast cell, and we also kind of stain for the T cell, because T cell is also immune cells that can get attached if there is no small molecule coating. But in our lead you can see there is no myofibroblast, then there is no immune cells. We also estimated like the collagen, because I said like fibrosis is basically combination of collagen and combination of myofibroblasts, right? Combination of collagen and combination of myofibroblasts. So we also estimated collagen, and as you can see compared to the control, all the leads have very low amount of collagen. So basically we identified these top two leads, right? And mostly we kind of selected this G4-110 as a lead to do the cell-based implantation in type 1 diabetes. So you can see in this figure what we did, we took human islet, and one thing to note that in this case we took a very high density islet, right? Why? Because a pancreas in our body contains lot of like almost like if I go few slides after, yeah. So each pancreas contains about like 1 million cells, right? So you can imagine like almost like 10 lakh cells. So each pancreas has almost like 10 lakhs of cells. So if we have to basically like in case of type 1 diabetes, as you know, like all the beta islet cells are destroyed. So basically we have to replace all those 1 million cells for a proper function, right? And to replace 1 million cells, lot more like capsules volume are required. Because you can imagine that if we can only use like 1000 cells per ml of capsules, how many liter of like basically the capsules will be required, and they can be implanted in our intraperitoneal belly area, but we don't have that much of volume in our belly area, so we cannot basically implant this high number of capsules. And then basically what is the solution? The solution is we have to increase the density of the islet. So you can see we introduce like a high density islet. These are all human islet. So we use like 4000 cell per ml, 8000 cell per ml and 16,000 cell per ml. Till this, before this work, the academic standard was to use only 1000 cell per ml. But that was too low, these were very low density. So we implanted like very high density cells, and as you can see like up to 3 months, and we did further like almost up to like 4 to 6 months, they could maintain the like normoglycemia level, they could correct the blood glucose. So whenever these active beta islet cells, these are all beta islet, these red cells are all beta islet cells present inside of the capsules, whenever they sense like a high glucose in the body, what they did, they secrete like insulin to the blood. And this insulin basically helped in terms of transporting the glucose, right? So they could maintain the normoglycemia level for a very long time, and even after explant you can see all the cells are alive, right? All the cells are alive. There are almost no fibrosis in the explanted capsules. So the lead in the last slide we showed you, right? So the lead Z1A10 was very efficient in terms of reducing the fibrosis in these implanted capsules and also like maintaining the normoglycemia. Now the problem is, as we said, that it takes about like a 1 million of islet cells to effectively implant or transplant in the body in order to basically have a proper cure for long term, because each like our pancreas has almost 1 million of pancreatic beta islet, almost 1 million of beta islet cells. Now to implant 1 million of beta islet cells using like a capsule based system, it will require like almost 4 to 5 liter volume, right? So it will require at least 4 to 5 liter volume of capsules, which is almost impossible to do, right? Which is almost impossible to think of. Because you can see, like in cases of any complication, what will happen, basically these capsules can get stuck in different part of our body and eventually it will become very difficult to retrieve those capsules. So what would be the primary goal eventually to develop? The main goal would be to develop a macro device or artificial pancreas. So it would be like a macro size object. There we can put this 1 million cells inside, and also in case of any complication we can easily retrieve the device and further load with new like alive pancreatic islet cells. So initially in this field, when like the type 1 diabetes therapy was started, first this Edmonton protocol was used where like the cells were directly injected, but as we said that host immune response will basically kill the cell. After that, this capsule based system were used as like a immunoisolation object, and small molecule coating also you can use, like we showed in the last paper in our work, where you can basically reduce the fibrosis response and can effectively cure the type 1 diabetes for 6 months to 1 year. But what we just said, that there are few challenges. First is like difficulty, there are to load high density islet, and even if you load high density islet, the volume becomes too much, so that in case of any complication you cannot retrieve those many capsules. Also the space is not enough to implant that many capsules. So eventual goal is to create like a macro device, but with a non-vascularized macro device, the device are unable to get proper nutrient support. So the final goal to create artificial pancreas is to create like a vascularized macro device, where there will be like nice vascular invasion or vascular network in between the device, and this blood network will basically carry the blood, and as you know, like blood carries oxygen and the nutrients. So as the blood carries all these required substances for the cells will stay alive for the long term. And this macro device also should be properly immunoisolated and if needed coated by the small molecule, so that our body's host immune response or the host like kind of fibrotic response cannot get triggered. Okay. There are different approaches for vascularization, but the most effective one is like transplanting like a vascular cells. So the eventual goal is like to create a certain design system where we have like a core shell type of structure. In the inner core part we will put like the beta islet cells. In the outer layer we will put like the vascularized device, which will have like a specific patterning, and this patterning will attract like faster vascularization. So you can see like in the outer shell layer we can have like different architecture pattern. It can be like serpentine, it can be even like parallel lumen, right? It can be like different octahedral type of structure, hexagonal and octahedral structure. It can be honeycomb structure. So all this pattern architecture actually stimulates faster vascularization. So any case of like pattern architecture, what it does, it stimulates faster vascularization, and faster vascularization basically helps in terms of delivery of oxygen and nutrient. So as you can see, a typical type of macro device or artificial pancreas can be developed with a serpentine structure that will have like lot of beta pancreatic islet cell inside, and after some time this device will actually form lot of vascularization around it, and this vascularization or blood supply will basically carry like oxygen, glucose and other ions, and all these inside islet cells will be able to stay alive and happy for longer time. So hopefully, like using this type of macro device, a artificial pancreas can be developed that can be eventually cure the type 1 diabetes for almost like 2 to 3 years. And in case of any complication, you can easily retrieve the macro device and further replace all the dead islet cells with a new fresh cell and again implant it to the body. So hopefully you like how the pancreatic like artificial pancreas can be developed. There are other ways of pancreatic like artificial pancreas also, including like hybrid closed loop or fully closed loop or dual hormonal system. These are basically like mechanical device which has like continuous glucose monitoring system and with a hybrid kind of injection of insulin and glucagon depending on the glucose level of the body. This type of injectable system or the artificial pancreas also can be created. But as I said, there are a lot of like challenges, including like high cost. Apart from that, there can be insulin resistance, and all the injection of insulin can cause pain to the body, especially for the kid. So ideally the target would be to develop like a cell-based therapy. Ideally the target would be to develop like a cell-based therapy, right, with a vascularized macro device, with a vascularized macro device where we can implant the cells in the body and the whole device should be immuno-isolated so that our immune system should not able to recognize it and destroy it. So this is the some of the modern approaches what researchers are taking. Hopefully you enjoy the class. So let's think about it. What is vascularization and why it is important for cell-based therapy, and what do you mean by angiogenesis, and also in general can you tell what is cell-based therapy. You can also refer different books and also please go through all the references what has been referred in the slide. Hopefully you are enjoying the class of human physiology. If you have any further question, please drop us the question in email. Also you can ask us during the live sessions. Thank you again for attending this very interesting class of cell-based therapy and artificial pancreas. Hopefully you are enjoying the classes. Let's meet with another new class very soon with you. Thank you.